Hello, everyone. I'm Dr. Andrew Kaufman, and my friend Sayer Ji asked me if I could record a brief video about the so-called Delta variant of SARS-CoV-2. So I thought I would put this little presentation together and actually give you the background and explain how the genome sequencing works in the first place, because all of what they call variants are all basically just different results of doing what they call a genome sequencing. Now, I want to also preface this by saying that these variants have not been actually studied with clinical correlation. So in other words, they didn't follow a group of 100 people or 1,000 people with this particular variant that they could reliably detect and then follow their clinical outcomes. So most of the information about what they tell us, that a variant is more transmissible or more severe or less severe or anything like that, are based on computer simulations and are purely theoretical. Okay, with that being said, I am going to share my screen and give part of the presentation that I've given before, which focuses specifically on the genome sequencing. So hopefully you can see the slides. Okay, so I'm just going to start off with the basics about genetics, which is all based on DNA and RNA. And these are chemical compounds that are what they call polymers, meaning that they're like a necklace, with each bead being slightly different. And there are 4 different types of beads that you can use to make this sequence: adenine, thymine, guanosine, and cytosine. And they're also called bases. And you can see in the strand in the middle where there are 2 lines of boxes with the letters matching up, that shows the double-stranded DNA. And these letters are complementary. So you see the top one is AT, so a T always goes with an A, and the next one, GC, they always pair up together when the different strands come together. When we talk about RNA, it only has 1 strand, so this pairing is not a thing, but it also has the 4 bases, and they correspond with the 4 bases of DNA. So basically, the order of the bases or the letters make up the specific sequence that distinguishes what that DNA does and also distinguishes one organism from another if you have a long enough sequence. Okay. So here is a slide that talks about what you could call the central dogma of genetics. Now, this is proven not to be accurate, but this is the framework that they describe that all this happens. So I'll explain that basically there is a sequence of DNA, and DNA gets made into RNA, and that process is called transcription. And then the RNA can then leave the nucleus where the DNA is and go out into the cytoplasm, the rest of the cell. And when it's out there, it can code for a protein. And this type of RNA that does this is called messenger RNA. You can see the strand on the top. And it is basically coding for specific amino acids, which are the building blocks of proteins. So you can see there's methionine, phenylalanine, and lysine. Those are different amino acids. And each one of those is paired up with 3 of the bases in the RNA strand, and that makes the protein sequence. And this is called the central dogma of biology or genetics. Okay, so what is the genome? The genome is the genetic code or the sequence of letters for an entire organism. So everything that they have in all of their cells. So in humans, we have 23 strands of DNA called chromosomes. And of course, we have 2 of each, except for the sex chromosome that we might have 2 different ones or 2 of the same one. And so all of the chromosomes end to end, the entire sequence, that's called the human genome. And it's distinct from other organisms. Okay. So how do you sequence a genome? Well, with the human genome, they started with an actual human and then took the genetic material out of the human cells, out of the nucleus, which are the chromosomes, and then they broke those down and sequenced each one into the sequence of letters or bases. And this took quite a long time because they were using an earlier technique called Sanger sequencing, where they have to have big, long strands and sequence one base at a time. but this is how they did it. So the key or operative concept is that they actually took the genetic material just from a human and not from any other source. Okay, so is that how they sequence the genome of viruses? Well, of course not. They have a special procedure for that. So how do you sequence a virus? Well, you perform what's called an in silico genome sequence, which means that it takes place entirely in the mechanics and circuitry of a computer, right? In other words, it's not actually from an organism. And this is what they do to give us the so-called genome of a virus. So they use a technique called next generation sequencing. And this is a modern technique that allows the process to happen much faster, because instead of sequencing a very, very long strand, one base at a time, it chops it up into small pieces and it cuts it in a way that it knows exactly where it cuts it. And then it can sequence those smaller pieces all at the same time in parallel, which saves a lot of time, just like a parallel processor in your computer is faster than a serial processor. But if you look, this is from a company that makes the package of technology to do this procedure. Oops, I skipped a slide by accident here. So going back to that, you can see at the top, there are two pathways because this is talking specifically about viruses. And you see on the left, or the light green pathway, it says to purify the virus. And that really is the gold standard, because then you know that every piece of genetic material that you are sequencing is from that pure virus and not from anything else. Now, it gives another pathway where you have semi-pure or enriched virus particles. So this is like a partial purification where the genetic material from the virus would be by far the dominant genetic material in the sample. And so it could be distinguished from the other materials because they'd be in a much lower amount. Okay, so then you take the genetic material out, which in the case of the alleged coronavirus would be RNA. And you basically, what they do is select out small pieces only, and then they make a library from it, which is really a step that you don't need to understand, but just know that every little strand of RNA of a certain length or shorter is included in that library. And then it sequences the library. And then it puts those sequences together into a big, longer strand, which is a process called alignment. And that's what I'm going to focus on teaching you about today. And then at the end, they say they've discovered a virus. However, since it's only in a computer and we don't know the source of the genetic material, in other words, it didn't come from just a virus, it came from other things as well. So we would call this an in silico or a theoretical genome. Okay, so we have here, this is the paper from Nature, which is the first published or the index genome sequence that was placed on the computer databases that we now can pull up as the SARS-CoV-2 genome sequence. And it was done by these researchers. And so here's what they did. They took the lung fluid from only one suspected case of SARS-CoV-2. And of course, they said that it was from COVID-19 because of a PCR test, which we already know is not accurate. But only